Today we're looking at embryology, the third week of development. Okay, without further ado, let's look at the objectives of this video, and we look at what we are focusing on and the events of the third week of development. So the objectives for this lesson today is: number one, you describe the formation of the primitive streak. Number two, you describe gastrulation and the events in gastrulation. Number three, you explain the notochord process and the notochord formation. And number five, you describe neurulation. Let's get into this video. All right, so the third week is the significant week of gastrulation. What is gastrulation? Gastrulation is the process of formation of the three germ layers, which are the ectoderm, the endoderm, and the mesoderm. So you have the ectoderm, the mesoderm, and the endoderm. These are the three germ layers. So the process by which the bilaminar disc is converted into a trilaminar disc is what we call gastrulation. So in week 2, remember that there is a formation of a bilaminar disc, which is called the bilaminar disc. And the formation of a trilaminar disc from that bilaminar disc is what we term gastrulation. So gastrulation is the beginning of morphogenesis, which is the formation of the body form and organs. It consists of the formation of the primitive streak, the three germ layers, and the notochord. The embryo at this level is referred to as the gastrula. Now, the primitive streak is what gastrulation begins with. So gastrulation begins with the formation of the primitive streak on the surface of the epiblast. Now, at the beginning of the third week, an opacity appears caudally in the median plane of the epiblast. Now, this opacity is due to the proliferation of the epiblast cells. The primitive streak results from the proliferation and the movement of the epiblast cells to the median plane of the embryonic disc. Now, very soon we're going to look at what causes these epiblast cells that are tightly adhered together to start moving to the median plane and to form the primitive streak. All right, so now this is the bilaminar disc that is formed in week 2, and this is the cut from the superior view. So you cut here, and then this here is the epiblast, and this one that is yellow underneath here is the hypoblast. Now we're saying that on the surface of the epiblast, this is where the primitive streak is formed. All right. So now what causes the formation of the primitive streak is the expression of certain factors that are called fibroblast growth factor 8. What this fibroblast growth factor 8 does is that it inhibits a protein that holds epiblastic cells tightly together called E-cadherin. So E-cadherin is downregulated by fibroblast growth factor 8. When fibroblast growth factor 8 inhibits E-cadherin, what happens is that the cells of the epiblast, these cells, will start moving in the median plane. And then when they move in the median plane, they'll form this. This is what we call the primitive streak. Now, the primitive streak consists of the primitive node on its cranial end, and in the primitive node, it will consist of the primitive pit. And then you're going to have a primitive groove in the middle of the primitive streak. Right. So this is what I've been trying to explain. Cells are added to the caudal end, which is this one here, of the primitive streak, causing the streak to elongate. So it will start from here, and cells will continue being added to its caudal end, and it will be elongating cranially. Okay. So at its cranial end, proliferation occurs forming a primitive node, which is this guy here. This is a primitive node. A narrow groove, which is the primitive groove, develops in the primitive streak that is continuous with a small depression in the primitive node, which is called the primitive pit. Alright, now this is what we're saying, that cells of the epiblast migrate towards the primitive streak, and the inward movement is what we call invagination. So when the cells arrive in the region of the streak, they become flat-shaped and detach from the epiblast and slip underneath. This inward movement is what is called invagination. Cell migration and specification are controlled by fibroblast growth factor 8, which is denoted as FGF8, which is synthesized by streak cells themselves. Now, this FGF8 downregulates adherent protein that normally binds the epiblast cells together. And FGF8 then controls cell specification into the mesoderm by regulating brachyury expression. All right. So what happens now is that the cells of the epiblast will start migrating through the primitive streak underneath it. So these cells will go underneath the epiblast to a region between the epiblast and the hypoblast. What they'll start doing is that these cells will start replacing the hypoblast. When they replace the hypoblast will turn into the definitive endoderm. And they are also going to move in this region between the epiblast and the definitive endoderm and form the intraembryonic mesoderm. When this happens, the epiblast then changes name to the definitive ectoderm. So this that is demonstrated here is the movement of epiblast cells through the primitive streak into the space between the epiblast and the hypoblast. They first of all replace the cells of the hypoblast, transforming it into the endoderm, and fill the space between the epiblast and the endoderm, forming the mesoderm, intraembryonic mesoderm. And then the remaining cells of the epiblast will be transformed into the definitive ectoderm. So this is what happens. So this is the endoderm, this is the mesoderm, and this is the ectoderm. All these three layers of the trilaminar disc come from the epiblast of the bilaminar disc. So all three layers of the trilaminar disc of the embryo originate from the epiblast of the bilaminar disc. So this process is what we call gastrulation. So remember, gastrulation starts with the formation of an opacity on the epiblast called the primitive streak. So the primitive streak, and then cells will now start migrating underneath the epiblast using the primitive groove and replace the hypoblast, turn it into an endoderm, fill the space between the endoderm and the epiblast, form an intraembryonic mesoderm, and the epiblast will be transformed into the definitive ectoderm. This is the formation of the trilaminar disc. Alright, so this is what we were talking about. Once the cells have invaginated, some displace the hypoblast, creating the embryonic endoderm. Other cells come to lie between the epiblast and the newly created endoderm to form the mesoderm. Cells remaining in the epiblast then form the ectoderm. Therefore, the epiblast, through the process of gastrulation, is the source of all the germ layers. The cells in these layers will give rise to all of the tissues and organs of the embryo. This is the process of gastrulation, and it's very important to explain. Alright, so this just explains what we were talking about. Movement of cells, replacing the hypoblast, filling the layer between the endoderm and the epiblast, forming the mesoderm, and this layer becomes the This layer becomes the ectoderm. Now, have you seen these layers where there is no mesoderm and the ectoderm and the endoderm closely adhere? Caudally, this is called the Crook membrane. Crook membrane. Cranially, it is called the bucopharyngeal membrane. We're going to see it. Alright, same schematic diagram showing the transformation from the bilaminar disc to a trilaminar disc in gastrulation. Okay, this is the same illustration that we were talking about. Okay, now what is the fate of the primitive streak? So normally the primitive streak undergoes degenerative changes and disappears by the end of the 4th week. Now remnants of the primitive streak may persist. If it persists, it will give rise to a tumor that is called the sacrococcygeal teratoma. The sacrococcygeal teratoma. So know this, the primitive streak is supposed to degenerate by the 4th week. But if it doesn't degenerate, it gives rise to the sacrococcygeal teratoma. Sacrococcygeal teratomas are the most common tumors in newborns and have an incidence of approximately 1 in 35,000 newborns. And the most affected gender, about 80%, are the females. Okay. So that is gastrulation. Now, let's move on and talk about notochord formation. Notochord formation. How is the notochord formed? So notochord formation starts with prenotochordal cells invaginating in the primitive pit. So they move forward until they reach the precaudal plate. Now the precaudal plate develops into the buccopharyngeal membrane, which develops into the mouth, and as embryology goes forward. So the precaudal plate is a small circular area of columnar endodermal cells where the ectoderm and the endoderm are in contact. I'm going to show you this very soon using a diagram. The precaudal plate is the primordium of the oropharyngeal membrane, which is the buccopharyngeal membrane. This simply means that the buccopharyngeal membrane is formed from the precaudal plate. It's located at the future site of the oral cavity, which is the mouth. The notochordal process elongates by invagination of cells from the primitive pit. Let's look at the diagram. Okay, so what happens is that this is a primitive pit. This is a primitive pit here. And then this is the precaudal plate. The precaudal plate is what gives rise to the buccopharyngeal membrane. So cells of the ectoderm will start invaginating and moving through the primitive pit. When they invaginate, they will move at the region between the endoderm and the between the ectoderm and the endoderm. So here they are going to form what we call a notochordal process. This notochordal process that lies in the mesoderm will form a canal. So which will be called the notochordal canal. Okay. And then this notochordal canal is going to fuse with the endoderm underneath. So there will be communication between the amniotic cavity and the yolk sac. Okay. As you can see here, there's a communication between the amniotic cavity and the yolk sac. This communication is through a canal that we call the neoenteric canal. So this is how the notochord is formed. Migration of ectoderm cells to the primitive pit forms a process between the ectoderm and the endoderm. Alright, so this is just the literature. The flow of the notochordal process fuses with the underlying embryonic endoderm here. And then the fused layers gradually undergo degeneration, resulting in the formation of openings in the flow of the notochordal process. The primitive peak extends into the notochordal process, forming a notochordal canal. So when a notochordal process is formed by the invagination of cells between the ectoderm and the endoderm, this is the notochordal process. And then a canal forms, which is called the notochordal canal. This brings the amniotic cavity in communication with the yolk sac. So as you can see, there is communication here between the yolk sac here and the amniotic cavity. Okay, so the openings rapidly become confluent and the flow of the notochordal canal disappears. The remains of the notochordal process forms a flattened, grooved notochordal plate, which is this one here. So now, this is the notochordal plate that is formed. This notochordal plate is going to form a groove here, and it will form a circular canal here, which is called the notochord. So beginning at the cranial end of the embryo, the notochordal cells proliferate and the notochordal plate unfolds to form the notochord. So the notochordal plate here will now unfold to form this rounded feature here, which is called the notochord. The proximal part of the notochordal canal persists temporarily as the neuroenteric canal, which forms a transitory communication between the amniotic and the umbilical vesicle cavities. When development of the notochord is complete, the notochordal, which is the neuroenteric canal, normally obliterates or it disappears. The notochord becomes detached from the endoderm. So this is the endoderm. The notochord becomes detached, which becomes when the notochord becomes detached from the endoderm will now become a continuous layer once again. So now, question is, what is the importance of the notochord? So the notochord will induce the formation of the neural structures in the embryo. So it will induce neurulation. So this is the process. This is the process of how it happens. So there is invagination of cells between the ectoderm and the endoderm, forming the notochordal process. This notochordal process is going to develop a canal, which is called the notochordal canal. The notochordal canal will be continuous with the endoderm, and they will fuse, and this layer underneath will disappear, forming a formation between a continuation between the amniotic cavity and the yolk sac. Okay, and this remnant of the notochordal process is what we call the notochordal plate. Then the notochordal plate will be continuous with the endoderm. It will unfold and make a circular fold, which is called the notochord. When it unfolds, it detaches from the endoderm, and the endoderm once again becomes continuous. The notochord is what we call the axial mesoderm. Where the notochord is here, this is what we call the axial mesoderm. Immediately lateral to it, there is formation of what we call the paraaxial mesoderm. And the paraaxial mesoderm, from the paraaxial mesoderm laterally, you have the intermediate mesoderm. From the intermediate mesoderm, you've got the lateral plate mesoderm. From all those structures of the mesoderm, they are important features that arise. Okay, so the notochord extends from the oropharyngeal membrane to the primitive note. It degenerates as the bodies of the vertebrae form from the somites, but small portions of it persist as the nucleus pulposus of each intervertebral disc. The notochord functions as the primary inductor or the signaling center in the early embryo. It induces the overlying embryonic ectoderm to thicken and form the neural plate, the primordium of the central nervous system. So the notochord, the importance is that it signals and induces the formation of the nervous system, the central nervous system, which is made up of the brain and the spinal cord. Later on in this lesson, we're going to look at how these structures are formed. All right, now let's look at the formation of of the allantois. This is going to be very quick. The cloaca membrane is formed at the caudal end of the embryonic disc. This membrane, which is similar in structure to the bulk of a pharyngeal membrane, consists of tightly adherent endoderm and ectoderm cells with no intervening mesoderm. So at the bulk of a pharyngeal membrane, there is no mesoderm. There is just endoderm and ectoderm that are tightly adhered to each other. When the cloaca membrane appears, the posterior wall of the yolk sac forms a small diverticulum that adheres into the connecting stalk. This diverticulum, which is known as the allantois, appears around the 16th day of development. Although in some lower vertebrates, the allantois serves as a reservoir for excretion products of the renal systems. In humans, it remains useless, but may be involved in the abnormalities of the development of the bladder. The proximal part of the original allantoic diverticulum exists throughout much of development as a stalk that is called the urachus. Now, what is the importance of the urachus? The urachus extends from the bladder to the umbilical region, and the blood vessels of the allantoic stalk become the umbilical arteries. So this is the importance of the allantoic stalk. The blood vessels that develop in the allantoic stalk become the umbilical arteries. You know, the umbilical arteries facilitate exchange between the mother and the embryo. All right. Now let's look at the clinical correlates of gastrulation. Teratogenesis associated with gastrulation. What could go wrong if gastrulation doesn't happen properly? Okay. So if gastrulation doesn't happen properly, for example, if the mother exposed to high doses of alcohol at the stage of gastrulation, it may result in the deficiency of the midline. Okay. Deficiency of the midline in the craniofacial structures and results in a condition that we call holoprosencephaly. Now, holoprosencephaly is the failure of the prosencephalon, which is the forebrain, to develop normally. So this condition may ensue if gastrulation doesn't happen properly. Okay, and the two lateral ventricles often merge into a single ventricle, and the eyes are very close together in a condition that is called hypothyroidism. So this is one of the conditions that may occur if gastrulation doesn't happen properly or effectively. Okay, and another thing that may happen is what we call caudal dysgenesis, which is also called serenomelia, which results from insufficient mesoderm that is formed in the caudamost region of the embryo. This abnormality includes hypoplasia and fusion of the lower limbs, vertebral abnormalities, renal agenesis, imperforate anus, and abnormalities of the genital organs. So in humans, these conditions, caudal dysgenesis, is associated with maternal diabetes and other conditions. Let's look at the picture of this. So this is what is called serenomelia, caudal dysgenesis. As you can see, the limbs are fused here because of insufficient mesoderm at the caudamost end of the disc. This we already looked at, which are tumors associated with gastrulation, sacrococcygeal teratoma, okay, which commonly contain tissues derived from all three layers. So we said this occurs because of the persistence of the primitive streak. All right, other birth defects are situs inversus, which refers to a condition where the positioning of organs is reversed in a mirror image arrangement. This condition includes dextrocardia. So dextrocardia is a condition where the heart is located on the right hemithorax instead of the usual left side. There's also what we call situs ambiguous or heterotaxy, which refers to discordant organ positioning with respect to symmetry, where one or more organs are abnormally reversed in position, or isomerisms. All right. So this is situs solitus, and then this is heterotaxy. So where organs are not correctly placed according to symmetry. And then this is situs inversus, where all your organs are placed in opposite directions. So the heart normally is supposed to be here, it's on the right. The liver is supposed to be this side, it's this side on the left. The stomach is on the left, and the spleen as well is on the right. The stomach is on the right, and the spleen is also on the right. Okay. Now let's look at further development of the trophoblast that happens in week 3 of development. So by the beginning of the 3rd week, the trophoblast is characterized by primary villi that consist of cytotrophoblastic core covered by syncytial layer. So what happens is that these primary villi will now be infiltrated by mesoderm cells. These mesoderm cells are from the extraembryonic mesoderm. Okay, and when they are penetrated by the extraembryonic mesoderm cells, they form the secondary villus. Then these secondary villus, the mesoderm in the secondary villus, will now differentiate into blood vessels and small blood vessels and blood cells, forming the tertiary or the definitive placental villus. So this is how the placenta is formed. So these are the primary villi. They only have cytotrophoblastic cells and syncytial cells. And then when this extraembryonic mesoderm penetrates the primary villus, they become secondary villus. When this extraembryonic mesoderm that penetrates the primary villus differentiates into small blood vessels and blood cells, then it becomes a definitive placental villus. So this is the primary villus. It only has the cytotrophoblast and the syncytial trophoblast. Secondary villus has got cytotrophoblast, syncytial trophoblast, and on the center it has mesoderm cells, the mesoderm cells. In the tertiary villus, the mesoderm cells differentiate into small blood vessels and blood cells. So this is the development that happens. Okay. So now this is the literature. Capillaries in the tertiary villi make contact with capillaries developing in the mesoderm of the chorionic plate in the connective stalk and in the connective stalk. These vessels in turn establish contact with the intraembryonic circulation system connecting the placenta and the embryo. Hence, when the heart starts to beat in the 4th week of development, the villus system is ready to supply the embryo proper with essential nutrients and oxygen. Meanwhile, the cytotrophoblast cells in the villi penetrate progressively into the overlying syncytium until they reach the maternal endometrium. Okay, so now this is what happens. It's saying that these cells and blood vessels that develop in the extraembryonic mesoderm of the placental villus will come into contact with the blood vessels that are in the connective stalk, connecting stalk. Hence, they'll be in contact with the blood vessels that are in the embryo here. So there is communication between the placenta that is forming and the embryo circulation. So when the heart begins to beat in the 4th week here, there is going to be circulation from the embryo to the placenta. And there is proliferation of these cells that are light green here, which are the cytotrophoblast, and they go and infiltrate the uterus here, so that there is establishment of blood communication between the uteroplacental, there's between the uterus and the placenta. Okay, so there is going to be invasion of the endometrium of the mother by the proliferating cytotrophoblast cells, so that there is communication between the blood vessels that are in the endometrium of the mother with the blood vessels that are forming in the placenta here. Let me just show you a picture. Okay, so this is the picture. As you can see, this is the blood vessels that are formed in the villus. Okay, so this is the placental villus, or the tertiary villus. These between these tertiary villus, there are spaces that are known as the intervillus spaces, and the proliferating cytotrophoblasts that are going into the endometrium, infiltrating the endometrium, are going to come into contact with the maternal vessels. These maternal vessels will infiltrate the intervillus spaces. And then these intervillus spaces now are going to form a communication between the placental blood vessels and the maternal blood vessels. That is how uteroplacenta circulation is established. Okay. Now, let's look at the last part of this lesson, which is neurulation. Neurulation is simply the process by which the neural tube is formed. The stages of neurulation include the formation of the neural plate, the neural groove, the neural folds and their fusion, neural crest cells, and the neural tube. So it begins during the early part of the fourth week, which is about day 22 to day 23, and ends by the end of the fourth week, which is day 27, and is induced by the notochord. Remember what we said. Neurulation is always induced by the notochord. So this is what happens. So the notochord, which is here, will induce the ectodermal cells here to thicken. When they thicken, they form what we call the neural plate. Then this neural plate will form the neural fold, the neural groove. So the underlying effect of the developing notochord, the underlying ectodermal cells thickens to form the neural plate. So just on top of the notochord, there's a neural plate that is formed. Okay, beautiful. Now the neural plate first appears cranial to the primitive node, which is here. So cranial to it, this is the neural plate, and dorsal to the developing notochord, which is on top, because you know that the notochord is inside, underneath the ectoderm. So just on top of the notochordal process, that's where the neural plate develops. So as the notochord forms and elongates, the embryonic disc elongates and becomes club shaped. So this is the neural plate. The neural plate broadens and extends cranially as far as the oropharyngeal membrane, which is here, and later grows beyond it. So as you can see, as the notochordal process is elongating, the neural plate is also elongating. The neural plate will then form folds, which are known as neural folds. So on the 18th day, the neural plate invaginates to form the neural groove and folds. So it will invaginate, meaning it will form a groove like this. And on top of the groove, you have the neural folds. The neural folds. So as the neural plate lengthens, its lateral edges elevate to form the neural folds, and the depressed mid region forms the neural groove. Neural folds, neural groove. So it's just of the notochord. So some neuroectodermal cells along the crest of the neural fold differentiates as the neural crest cells. So on top of the neural fold here, there are cells, neural crest cells that will form. Okay, neural crest cells that will form. And as you can see, the neural groove keeps on enfolding, and eventually it is going to form the neural tube. Okay. So by the end of the third week, the neural grooves move to the midline and fuse to form the neural tube. So these will move to the midline like this, and they'll form the neural tube. The fusion begins in the future cervical region and then extends both in the cranial and in the caudal direction. This is what happens. This neural fold, the neural groove, will now unfold and form this neural tube. Okay. The neural tube. So the neural tube, when the neural tube forms, you have an anterior neural pole and the posterior neural pole. So this is the formation of the neural tube. So the neural tube separates from the surface ectoderm. It lies in the midline and dorsal to the notochord. So this is the dorsal notochord here. Okay, the notochord was here. So as the neural tube separates from the surface ectoderm, neural crest cells form a flattened irregular mass, the neural crest. This is the neural crest between the neural tube and the overlying surface ectoderm. The neural crest soon separates into right and left parts. So this neural crest will separate into the right and the left parts that shift to the dorsolateral aspects of the neural tube. Here they give rise to the sensory ganglia of the spinal and cranial nerves. So where there's ganglia, the ganglia is where cell bodies are found. These come from the neural crest. So neural crest cells subsequently move both into and over the surface somite. Okay. So these are the derivatives that come from neural crests. So the connective tissue and bones of the face and the skull come from the neural crest. The cranial nerve ganglia, the senile cells of the thyroid, the conotruncal septum in the heart, the odontoblasts, the dermis in the face and the neck, the spinal ganglia, sympathetic chain and pre-aortic ganglia, parasympathetic ganglia of the gastrointestinal tract, adrenal medulla, Schwann cells, glial cells, meninges of the forebrain, melanocytes, smooth muscle cells to blood vessels of the face and the forebrain. All of these come from the neural crest. Okay. So the neural tube is open at both ends, communicating freely with the amniotic cavity. So the cranial opening, the anterior neural pole closes at the 25th day, and the caudal neural pole closes at the 27th day. Neurulation is then complete. So the central nervous system is represented by a closed tubular structure with a narrow caudal portion, the spinal cord, and the much broader cephalic portion. This is a cephalic portion, this is a caudal portion. So we're saying the caudal portion becomes the spinal cord, the cephalic portion becomes the brain. Okay. So now the brain, the cranial 1/3 of the neural tube represents the future brain, this one here. And the caudal 2/3 represents the spinal cord, which is this one here. Alright, so this is how neurulation happens. I hope you've learned how gastrulation happens, how the allantois is formed, how the placenta, uteroplacenta circulation is established, and how neurulation happens. Thank you and see you in the next video.